Alright, welcome back. So we did macromolecules part 1. Now we're going to pick up with proteins and the nucleic acid. So proteins, we can see here, it's kind of the beauty category. Skin, hair, nails, muscles. Those are all, their main component is protein. So the monomer of a protein is called an amino acid. Elements include carbon, hydrogen, oxygen, and nitrogen. In some amino acids, sulfur is also a component, but it's not in all amino acids. The bond between amino acids when building a polypeptide is called a peptide bond. And the polymer is a polypeptide or a protein. Okay, you'll have those words interchanged. Proteins function in our body in many ways. So it is our primary structural component. So when you think about tissues and muscles and things like that, that's protein. They're primarily composed of protein. However, also within our bodies, we have these little worker helper molecules called enzymes. Without enzymes, our bodies would not be able to function. We would not be able to be multicellular without the presence of these enzymes. And so these enzymes help reactions to take place in an efficient manner. And so here we're looking at 2 amino acids. Again, we have dehydration synthesis because we are losing water in order to build a polypeptide chain. And so all amino acids have these 3 parts. So they all have an amino group. So remember back from our functional groups that we studied, they all have an amine group. They all have a carboxyl group, so that's where we get amino acid because the carboxyl is acidic. There's a hydrogen, and then the center carbon is also bonded to some other functional group. This other part here kind of determines how this amino acid is going to respond and bond with other amino acids. There are 20 different amino acids. And so we're going to talk about how proteins are put together and why their bonding is so important. The initial sequence, we call it the primary structure, just kind of, amino acid 1, 2, 3, 4, 5, whatever amino acid comes after the next. That order is important because it does affect all the other folding and conformation and job that protein will have. Okay, so we're going to talk about these 4 levels of protein structure. So the primary structure, again, is just the sequence. So if you have taken a look in your book at the types of amino acids, like this one might be glycine, and then we have valine, and phenylalanine, and leucine, and isoleucine, and whatever. But it's just the simple chain of amino acids, and the bond between them is a peptide bond. Okay? So if you want to think about it, I might put a video up in a little while about the structure. It's like the edge of a piece of paper. It's just very one-dimensional, non-interesting. It's just, 1, 2, 3, 4, 5. The secondary structure, however, we start to get a little bit more dimension. Here we can see hydrogen bonding interacting between the different amino acids. And that hydrogen bonding can cause 2 forms, or 2 structures. Either the form that we see here called a beta-pleated sheet, or it could form an alpha helix. building up from that, we have the tertiary structure. And so this tertiary structure is really influenced by hydrophobic and hydrophilic interactions. So amino acids that are hydrophobic will dive to the interior of this structure and cause folding, while the amino acids that are hydrophilic will want to be at the surface of the protein. And this makes a protein that has a final 3D shape. This could be the final step of a functional protein. Some proteins work with others though, and so here we have several tertiary structures put together into a quaternary structure, and so this example is hemoglobin. So hemoglobin is composed of 4 different polypeptides. And so for hemoglobin, for this enzyme to work properly, all 4 of these polypeptides have to be composed and folded properly in order for this entire molecule to work. And so with this whole process, whatever final shape that protein has a very, very big impact on its function. If for some reason it were to lose its shape, that is called to be denatured. And it can either reduce the protein's efficiency at carrying out its job or make it completely non-functional. And so proteins have an optimum temperature and pH range that each protein works best at. So for example, the proteins in your stomach that digest food, those work best in an acidic environment. However, the proteins that are in your blood for the transport of oxygen, those work at a more neutral pH. And so if the pHs of those areas were to flip-flop, neither protein, neither enzyme would be able to do its job. And so we mentioned enzymes earlier, that they are molecules that help catalyze reactions, that they're really what make your body efficient and able to function. There are lots and lots of proteins. These guys, chaperone proteins, kind of help make sure that a protein is folded properly. When these chaperone proteins misbehave or don't do their job properly, we think that it could be linked to some other conditions such as Alzheimer's. And so here we're looking at DNA. So this wild type is normal DNA. And then we're looking over here at a mutant DNA. We can see that this A has replaced the original thymine. And so when that is transcribed into RNA, we can see that there's a change. And when it's translated into the protein, instead of having glutamine, we get valine. And so that has a very big impact on the function of this hemoglobin. Instead of being a nice round cell, now we get a sickled cell. And so sickle cell anemia is one condition where we can show that where a protein's misfolding can impact its job. So this protein, this hemoglobin that's misfolded, can have an overall impact on the cell's shape, and it does not carry oxygen or carbon dioxide as efficiently as a normal round donut-shaped cell. Also, these sickled cells can get clogged in capillaries, causing a lot of pain in extremities, the hands and feet in particular. And I know in the U.S. it is primarily our African American population that is impacted by sickle cell anemia. And that's basically an indicator of their ancestral origin. And so it was really an adaptation to prevent the spread of another disease called malaria. And so malaria is frequent in these areas of Africa and Asia. And so although we did mention specifically African Americans, it does also affect people of Asian descent as well. And so you can see in these regions of high instances of malaria that the people are also more likely to carry the sickle cell anemia gene. And that's basically because this malaria is a blood-borne pathogen that does not like sickled cells. It does not find a good host in a sickled cell. And so the people who have sickle cell anemia are resistant to malaria, and so those people are able to grow up and have kids and pass on their genes to the next generation, passing on that sickle cell trait. Whereas the people who didn't have that sickled cell, just had normal red blood cells, well, they are extremely prone to the catching of malaria and then dying at a young age and not having kids and not being able to pass on their genes. So protein, most people eat, meat, poultry, fish, whatever, to get protein. But if you're a vegetarian, most cultures have a bean and grain combo that they eat. And by eating a bean with a grain, you get all 20 amino acids. And so like in Asian countries, having soybeans and rice, that gives you a complete protein. Eating red beans and rice, that gives you complete protein. So as long as you have a grain, like corn, with a bean, a legume, then you're fine. So try this question. And hopefully you got it right. Peptide. Alright. This one's a little tricky. Think root words. So protein should automatically tell you has a protein. But what about glyco? There you go. Alrighty, last group, nucleic acids. So these are DNA and RNA. So nucleic acids have a monomer called a nucleotide. This is a very complex monomer. It's actually a 3-part monomer. It has a phosphate group, a pentose sugar, and a nitrogen base. The elements involved are carbon, hydrogen, oxygen, nitrogen, and phosphorus. The bonds between each nucleotide, each monomer, it has 2. When forming just the up and down chain, those are called phosphodiester bonds. But when we're looking at DNA and it's bonding to its neighboring chain, that's a hydrogen bond that's joining those. And so the polymers can be either DNA or RNA. And that should say polynucleotide. And in our bodies, this stores our genetic information. And so the nitrogen bases, we have adenine, thymine, cytosine, and I believe guanine is cut off on your screen at the bottom. So again, the phosphodiester bonds are the ones between the phosphate going up and down to each sugar. And then the hydrogen bonds are the ones in the center, represented by the dots. And we're going to talk lots and lots and lots and lots and lots about DNA and RNA. And so we said that nucleic acids store our genetic information, and so the DNA has our genes. You might be familiar with this word genotype, and that gene has information that tells your bodies how to make a protein, or your phenotype, your physical appearance. You should remember that adenine goes with thymine, cytosine goes with guanine. If you need a hint to remember, A-T, apples grow on trees, and C with G, cars go in a garage. So think of root words on this one also. Hopefully you got it. Alright guys, so good job. I know we've been through a lot of material in a short amount of time. There is a macromolecule quiz coming up soon, so please be sure that you are reading, that you've watched the videos, that you've gone over your notes, and that you are ready to go for quiz time. Alright, see you guys soon. Bye.